We're in. Yes. All right, so. How do I get rid of all this nonsense up here? Okay, so does everyone know the difference between innate and adaptive immune? We talked about this. Pretty sure we talked about this. So innate and natural, you're born with. So your body really doesn't have to do a whole lot about it. Adaptive, specific, and must be acquired, like with COVID-19. If you have antibodies to it, that you adapt to that. All right, you weren't born with that. You're exposed to whatever it is, if it's COVID-19 or any other pathogen, you have to develop antibodies to it. I know we talked about antibodies before, all right? But your first line of defense is innate. So these are things you're born with. We talked about physical barriers, skin. Do you guys remember anything about tears? What's in tears? No, you guys remember?
IG.
You guys remember gram-positive bacteria at all? Okay. And how did penicillin work?
By the cell wall.
All right, cell wall. So your tears actually have lysosomes in there that actually break down the cell wall of gram-positive bacteria, same as penicillin. So tears, they have lysosomes in them, sneezing, chemical barriers, low pH. Where do we have low pH? Anybody remember? So it'd be very acidic, low pH.
Your stomach.
Skin. All right, in your stomach. We have lysosomes in your sweat, your tears. All right. Do you guys remember anything about white blood cells? The granulocytes? Family?
Histamines in them.
Okay, they have granules in them. They have lysosomes in there. All right, and we're going to talk today about peroxide. How it can oxidize the bacteria. And we said that some bacteria can neutralize oxygen. It can deal with that. But if we give it a respiratory burst of a lot of oxygen, it can't deal with all that. Right? So do you guys, have you been using your hand sanitizer recently? Okay.
No, I just washed my hands.
So I made some industrial strength stuff. So if you need some, well, you can't get it in camps
Oh,
anyway.
God.
I made some with 80% alcohol, 30% peroxide, and glycerin. All right. So this shit will eat through anything. All right. But it's got peroxide in it. All right. So there's no bacteria or virus that can deal with that amount of oxygen from the peroxide. We talked about digestive enzymes before. We have some genetic barriers, and you probably know people that genetically get sick a lot more often, or some families that are genetically, their immune system may not be as fit as others, and that might be genetics. And we know that some diseases or pathologies have genetic propensity for some races than others, right? And we'll talk about that. Your second line of defense, once it breaches your first line of defense, which is all of your skin and everything we talked about, your mucous membranes last week, once it gets breached through that, we have our second line of defense, all right? So phagocytosis, cell eating, inflammation, fever, and interferon. Now, I know we talked about this last week. Does anybody remember about interferon? This is a chemical that your body will send out to other cells that are infected with the virus. We talked about apoptosis. These cells are going to have programmed cell death, so they don't become virus factories. Right? And then our third line of defense we'll talk about in chapter 16. All right, this is acquired, very specific, and we'll talk in great detail about your T lymphocytes, your B lymphocytes, and antibodies. Right. So remind me if I haven't talked about something. We've already gone over in pathophys, so I might get a little confused, but I know I've mentioned it so far this semester, at least briefly. Right? These you guys can watch on your own. The other videos I've embedded will not work on my MacBook Pro. They work on my old laptop they gave me when I first started like 6 years ago, but it won't open WebEx. So I think I have to throw it in the garbage. And my notebook that the school gave me won't open up. I can't even sign on to it. So this is what I'm stuck with right now. Right. So we'll talk about defense mechanisms. All right, first line of defense, we just briefly said any barrier that blocks invasion of portal of entry. We talked about your hand sanitizer and all the alcohol in that. And does anybody remember why I said that's not a great idea to use it a lot? Nobody? I see this is not going to be interactive. Okay. All right. So anyway, all that alcohol is gonna destroy all of the oil in your skin. It'll start cracking your skin. All right. And then bacteria has a way of getting in past your first line of defense. And remember, past your basement membrane is your capillary bed. Once anything gets into your capillary bed, it becomes the stomach. All right. So hand sanitizer is great if you are exposed to something nasty, or if you know you're going to be across something, it's great, but it wouldn't be something I would indiscriminately use. I know in a lot of healthcare settings you have to use it, especially going from patient room to patient room. Your second line of defense, well, we'll talk about that today, protective cells and fluids, inflammation, phagocytosis, all right? And does anybody remember our general protective cells that don't need a special introduction? We're talking about PAMPs, which is pathogen-associated molecular patterns. All right, so your phagocytes have, they can recognize general patterns in bacteria, viruses, worms, and things, just some general patterns that a lot of pathogens have. They're not very specific, like your third line of defense, but they do sort of work. Right? You guys are too young to remember. Well, did anyone hear of a Jeep? An older Jeep? So they're double-sided keys, all right? So a lot of these keys back in the 70s and 80s were double-sided, and I said before, I remember I mentioned in class that Ford and Chevy and Chrysler used to make like 10 or 15 keys. So you could literally grab a key for your car and go in a big parking lot, and that key would start many, many different cars, get you into many, many cars. All right, so that's similar to these PAMPs. They're very, very specific. They just kind of have a general idea what these pathogens look like, and they go in and attack it. And start, if you're a monocyte or a dendritic cell, you're going to actually go in and process this. You're going to present the antigens to your immune system. The immune system will make very specific antibodies to that. And your third line of defense with your T lymphocytes and your B cells will start making antibodies to very specific pathogens. All right, so here's just, I know we talked about this before earlier. So your physical anatomical barriers are your first line of defense. So your skin, we talked about tears, lysomes up here, fatty acids, saliva in your mouth, and your tears. Once again, I said that they can go and they can start lysing a lot of different bacteria. On your skin, you guys unfortunately can't do your skin lab and micro, but if you were to swab your skin, if we didn't have mannitol salt agar, anything would be growing. You could have mold spores in your skin, or E. coli or Proteus, things like that would be growing. But if we use a selective media, it would only allow things that can live in very high salt environments, and that would be your staph. Right? So realize your skin is got some lysomes in it. It's very oily. It can be very, very salty. It tends to retard the growth of a lot of different things. All right, but once again, you want some things growing in your skin. You want a basic microbiome there, so that really pathogenic things don't have room to grow, and they're competing with other things that are probably less pathogenic. Right? And stomach acid, we talked about earlier, that's very, very low pH, very, very acidic, will kill most things. Like if you're coughing up or you're sick and you either blow your nose or spit it out, or spit it on your sister, or whatever you want to do with it, or swallow it. Hopefully the stomach acid in your skin will, or stomach acid in your stomach will kill most of that bacteria. But later on, when we, next month, when we start talking about worms, remember these worms are going to be secreting cysts and eggs. And when you swallow these things under your fingernails or wherever, we'll get into the specifics of that. Remember, the stomach acid will start encasing or unencasing these eggs, and the larvae will start hatching in your stomach. They get your duodenum, and they start reproducing in your intestines. So that's a little yummy topic we have to talk about later. So here's your, hey Garrett, can you see me now? Is your camera working?
I can see you. I can just get, can you hear me?
Yeah, I can hear you.
Oh, yay.
All right, so it's working. All right. So here's your physical and anatomical barrier, your first line of defense. All right. So here's your cilia. All right, and we'll talk later on about some of these diseases that go in, they start destroying your cilia, causing major havoc in some of these diseases. Later on, we'll talk about that. And, you know, not to lecture you guys, but if anyone's smoking now, probably the worst time in history to be smoking, you're going to start killing that cilia, and you'll start compromising your type 2 pneumocytes, and we won't even get into what that can do with COVID, all right? But we're starting to find out that COVID is not really, I don't want to get into the details of it, but it's not as pneumogenic as we thought. It actually has to do with getting into your red blood cells and breaking down the heme. All right, and it's not really related that much to pneumonia because it's actually finding out it is bilateral in the lungs, not unilateral. So non-specific chemical defenders, sebaceous secretions from your skin, the oil, things, bacteria can't really get through that, or it doesn't really have a hospitable foundation for a lot of the bacteria, which is a good thing. Lysomes, we said before, lice. These lice bacteria hydrolyze the cell walls of bacteria like gram-positive bacteria, some gram-negative. Skin's very acidic, hydrochloric acid in the stomach, digestive juices, and the bile of the intestines. All right, and I know I mentioned this before. They probably told you in A&P that bile was strictly to emulsify fats, correct? Maybe you didn't hear that. All right, but a lot of bile inhibits a lot of gram-positive bacteria. So unfortunately, we're not going to be doing the Enterobacteriaceae lab either. Dr. Hoffman and I will try to put one together for you guys. But please realize the bile from your produced in your liver, stored in your gallbladder, when it's released, it emulsifies fats, but it also inhibits the growth of a lot of gram-positive bacteria. So you're going to find out in your intestines, the Enterobacteriaceae are mostly all, I would say 95% of them, for all intent and purposes, are gram-negative. Right? So bile will inhibit gram-positive growth in the intestine. Right? Oops, what happened? And then genetic defenses. Some hosts are genetically immune to diseases of other hosts. Now you guys are going to find it extremely hard to believe, but I literally have never had the flu. Right? And I literally do not remember ever being sick, other than a headache, ever in my life. Right?
Knock on wood.
Yeah, seriously.
Got it now. But my brothers don't get sick either. Right? My mother never got sick. Right? Some pathogens have great specificity. All right, whether they're zoonotic or not, they're very specific to certain genetic makeups and very specific to certain cells. Right? And like I said, some genetic differences exist in the susceptibility with different people. Right. We'll get into immunology. I know a lot of teachers tend to skip this, especially with A&P, and I know that some of the micro teachers tend to skip it and they'll give you a packet on it or whatever. I unfortunately think it's extremely important that you learn this if you're going to healthcare, right? And there's a lot of board questions usually on immunology. Right, so the study of bodies, second and third line defense is immunology. Functions of a healthy functioning immune system. So we want to survey the body. All right, we want to go to the body and check things out. All right, anybody remember what cells those are? Nothing. Okay. All right, so we want to recognize foreign material. And I said before, today we're talking about, right here it says PAMP, pathogen associated molecular pattern. So these are just really genetic. So, you know, as an example, if you were walking down the street, you would be able to tell if there's a dog, you might not know the breed of it, or a cat, or if somebody was purple or orange, you'd notice that they were different. All right, you wouldn't know specifics of it, but you would have a recognition pattern of something that just didn't seem right. Right? And you would recognize that, and these cells will go in and they'll engulf them, and they'll take some of the surface markers and they'll present them to your immune system and say, listen, I don't know what this is, but it's not normal. So, you know, here's my job. I'm presenting it to you. You guys figure out what antibodies you want to make to it. go talk to the B cells and see if they recognize it. If they do, they're going to make specific antibodies to it. All right. When we start talking about B cells, remember your body's going to have hundreds of thousands of B cells that are just waiting. They're called naive. They're just waiting to be exposed to something foreign. They have a lock and key to it. So your body will literally take all of your genetics. It'll start reshuffling it like the lottery, like a quick pick. And will make hundreds and hundreds of thousands of B cells that are just waiting their whole life. Maybe they'll be exposed to it or not, waiting for a specific antigen that they recognize, and they'll start making antibodies to it. Or they could spend their entire life walking around just waiting, living in your lymph nodes, just waiting for a foreign substance to be to recognize and start making antibodies to. But we'll talk about that. Hopefully we'll get started on that today in chapter 15. All right, and then their job is destruction of entities deemed to be foreign. All right. The problem is, we start talking about autoimmune diseases, sometimes your body misreads things as foreign. All right, and has anyone heard of the hygiene hypothesis? Does that sound familiar to anybody? All right, it's pretty big right now. They're saying right now that people that have all these allergies or a lot of these autoimmune diseases, literally, it sounds crazy, but your immune system's bored. It's used to having worms and ticks and fleas and all kinds of things like third world countries. So your immune system is just sitting around bored. It's waiting to find anything to irritate it so it can start waging a war on it. Right? Kind of like, does anybody have a sibling that just waits to instigate something? Very similar to that. Right? So your immune system is a large, complex, diffuse network of cells and fluid that penetrate into every organ and tissue. All right. Can anyone think of an area that wouldn't have immune function? Can you think of any area at all?
Cord?
Hair?
Cord?
Spinal cord. You have central nervous system. Yes. All right. So that is very few immune cells. All right. So do you guys remember anything about astrocytes or glial cells? Sound familiar at all? All right, so those are your immune cells of your central immune system, your central nervous system. They're different than the rest of your immune system, which is floating around in your interstitial fluid, all right, and your lymph nodes and your lymph vessels. So we have four major subdivisions of the immune system. We have reticuloendothelial system. So these are in between all of your cells in the capillary bed. We have these cells that are just sitting around waiting for something foreign to engulf and present to your immune system. We'll see a picture of that coming up. And your extracellular fluid or interstitial fluid. Remember, I said they're going to try to trick you on your boards. Remember, extracellular fluid is any fluid that's not in your cells. So that can be blood, that can be your joints, it can be extracellular fluid, it can be, remember third spacing? I think we talked about that. You have edema or your body's going to sequester fluid. That's extracellular. Anything that's not inside your cells is extracellular. But right now we're talking mostly about your interstitial fluid. This is the fluid that bathes all of your cells outside of your capillary bed. Remember, outside your capillary bed, you have that fluid, and you have your capillaries and your lymphatic system. And the point of that, I talked about hydrostatic pressure, non-contact pressure. Any of the fluid that doesn't quite make it back into your capillary bed is going to get into your lymphatic system. And where does your lymphatic system go to before it goes back to your thoracic duct or your right subclavian? Does anybody remember?
Heart?
Before it gets there, Garrett, it has to go through something that looks kind of like a kidney. It has to percolate through your lymph nodes, all right? And inside your lymph nodes are going to be your B cells and your T cells. So any of the fluid in your body has to go through your lymphatic system. And these vessels take it to your lymph nodes. And that's good because remember, we have a bunch of afferent vessels going into your lymph nodes. And then we have one efferent. So there's a bunch going in, one going out. It's kind of like a traffic jam on purpose. We want all these streets coming in. with one exit. So all this fluid has a chance to stay in your lymph nodes, percolate around, and your B cells and T cells get to literally bump into it and not more or less taste it, but it has to figure, it bumps into it. And if it recognizes it foreign, all right, we'll start waging a immune response to it. Right? And once again, if your B cells are naive to this, they're waiting their whole life to bump into something that'll agitate them, and they'll start making antibodies to it. Right? If it's your first exposure, anybody remember how many days it takes for that first antibody to be produced? It can take, anywhere from three days to a week. But the cool thing is, once we're exposed to that, we have those memory cells to it. So if we're exposed to it again, kind of the theory behind vaccination, if we're exposed to it again, we wage that immune response immediately to it. All right, so that's a good thing. We're exposed to it, a prior exposure to it. And we'll see that in a curve coming up. Okay, this is not going to work. It should play if you guys have Windows PC, and I think I've embedded the videos in Blackboard for you. Did anybody see the Gumby video? Nobody saw my endocytosis video that I made?
Just that you posted it.
You didn't see it?
Oh, not yet. I watch your videos later.
I tube chopped that. All right, you guys should watch it. So that was the cartoons when I was a kid. It was made in 1958. All right, I wasn't born yet, but that's what we had back then, claymation. So that video clip, that probably took them a month to make that little clip. All right. Anyway, so your immune system definitions, your white blood cells, your leukocytes. Leuko means white. What's the difference between a site and a blast? Do you guys remember? Sites are your grown-ass cells. Your blasts are your baby cells. Remember? No? Okay. So innate capacity to recognize and differentiate any foreign material. All right, and we'll go over your complete blood counts, what's elevated, what it means. All right, non-self is anything foreign. That can be anything. And once again, when we talk about chapter 16 or 17, your autoimmune diseases, sometimes your body misreads things, or we're going to talk about sequestered cells. So there's some areas of your body that the cells are, your immune system's never exposed to it, right? One is in the back of your eye. The other one, there's a few in the reproductive system in males and females that are sequestered. Your immune system never comes in contact with those. So if there's a problem, like let's say that you have an injury to your eye, if your vitreous humor in the back of your eye, if your immune system's exposed to it, it will find that as foreign. It'll start attacking it. So a lot of times if you have an issue with an eye, you have something going on, a lot of times they will enucleate your eye. They'll take it out because they don't want your immune system waging war against your vitreous humor. So what will happen is if they don't take it out, your immune system will start attacking the other eye. So you have to lose one eye to save the other, unfortunately. Beyond the scope of the course, but just so you heard it. All right, self, normal cells of the body. And then today we're going to talk about, once again, these pathogen-associated patterns, PAMPs, molecules shared by microorganisms. So if it is general type of bacteria, your immune system won't know exactly what it is, but it'll know it's foreign and it'll wage a basic attack against it. Pathogen recognizing receptors, receptors on white blood cells for pathogen-associated molecular patterns. And this is what your white cells, their little receptors or feelers, they go around and they just kind of notice something's odd. They go on and capture it. Endocytosis, like the Gumby video. All right, you can watch that. And it'll engulf it, process it, wraps it up nice and pretty, like a Cone 5 pottery package. If you've ever been there, they package things really nice. Presents it to your immune system and says, listen, I found this, it's foreign. Now you go find this marker and I need you to wage attack against it. So here is your reticuloendothelial system. So once again, here's your capillary bed here. Can you guys see the mouse?
Mm-hmm.
All right. So here's your capillary bed, right? So we talked about diapoesis. Right? So PEDI, what does that mean? Pedal, foot, diaphoresis. So these will actually, your capillary bed will open up, and these white blood cells or your immune system cells will leave your capillary bed. They'll go into this extracellular fluid, right? And right here is a white blood cell. It's just waiting to come across something foreign. Right? So as this fluid comes out here, the pressure builds. If it doesn't, the fluid doesn't go back in the capillary bed, the pressure here builds. It opens up your lymphatic capillaries. Right? They open up. When the pressure equalizes, these valves shut. And this fluid, as you squeeze your muscles, the fluid goes to your lymph nodes. It percolates through there, like a coffee, percolator, percolating through there, makes its way out. And once again, your immune system is waiting for foreign invaders, recognize them, and wage the attack against them. Right? And then, as Garrett said, eventually it's going to go back to your heart and recirculate. It has to go through. On the left, it goes to your thoracic duct. On the right, it goes to your right subclavian. And it goes back to your heart and recirculates. So here's your reticuloendothelial system right here. All right. Inhabited by phagocytic cells. So phagocytes, phago means eat. These are adult cells that eat. And the mononuclear phagocyte system, macrophages are ready to attack and ingest microbes that pass the first line of defense. So this is second line of defense. It already breached, somehow got in your body, got past your skin, got through digestive tract, it somehow got into your skin, into the extracellular fluid, and now we have our second line of defense. It's going to attack it and process it. All right, we'll talk a little bit here about blood. I'm sure hopefully this is all review for all of you. So your whole blood consists of plasma-informed elements. Serum is a liquid portion of blood after a clot happens, minus the clotting factors. So in school, we order defibrillated blood. So we take all the fibrin out of it. So that blood, when we take it out of the bottle, will not clot on the plate, right? So let me ask you guys something really quick, not on your test, but should your blood clot outside of your capillary bed? The answer is yes. The minute your blood is outside your capillary bed, it should clot. Your blood should never clot in your capillary bed. Right? So if you have intravascular coagulation, right, that's caused by LPS. And I talked about gram-negative cells before. We said LPS causes intravascular coagulation. It is a virulence factor released by gram-negative bacteria. It'll release that upon death. And that LPS will go in and it'll start causing your blood to coagulate in your capillary bed, causing petechia, those little pinpoint red spots that I've showed you guys before. But anyway, your blood should never clot inside your blood vessels. It should always clot the minute it's outside your blood vessel. So if you get cut and you nick your capillary bed, that blood leaves your capillary bed, it should clot, unless you have hemophilia, or you're deficient in factor 7, or you have Wildebrandt factor, or whatever it is. They're not outside of the scope of the course, but hemopoiesis. We all heard of this? You can nod, maybe no. Production of blood cells regulated by your kidneys. All right. Stem cells are undifferentiated cells, a precursor for new blood cells. All right, so if you have a stem cell, it should be able to make any blood cell at all. White thrombocytes, plasma cells, it should be able to make platelets. Leukocytes, leuko is white, cyte means big or adult, so these are white blood cells. Granulocytes, our lobe nucleus. We're talking about granulocytes. These are anything that ends in PHIL, the Phil family. We're talking about the bloodline, the stem of blood cells. And your granulocytes have granules in them, all right? And we said before, we have lysomes. So these cells will actually go in, engulf, or attack other cells, and they literally hit them with granules, all right? So when those granules are kind of be digestive enzymes or peroxide. So if we're talking about eosinophils, ends in PHIL. It has granules. Those will go in and they'll attach to a worm or something large, parasitic infection, and they'll keep hitting it with peroxides and they'll start destroying that worm or whatever that big pathogen is. Agranulocytes, A means none or no. The unlobe around the nucleus. All right, so those are anything that doesn't end in PHIL. So, and A&P or Patho or any of your books, you're going to have something similar to this. So the stem cells might be on the left and they go to the right here. They happen to be here. So just review your different stem cells. So your lymphoblast, monoblast, your myoblast. So you guys have heard of cancer or leukemia, right? So there's lymphoblastic and myoblastic. So they're two different stem lines. All right, so myeloid stem line is going to make everything, your red blood cells, your megakaryoblast, which are your platelets, and any of your granulocytes. So if you don't memorize that, if you remember your lymphoblasts or your lymphoid stem cells, they make all of your T cells, B cells, and your natural killer cells. So memorize both or at least one of them. If it's not from the lymphoid, it comes from the myeloid stem line. All right, so there's only two stem lines. Memorize, most people learn the lymphoid. Your lymphatic system, remember, is your immune system. And that's your T cells and B cells. All right, so if you want to memorize that and your natural killer cells, everything else comes from the other stem line. So I know earlier in the chapter, earlier this semester, I wrote out a CBC with differential and I wrote down never let monkeys eat bananas or wherever you guys learned it. All right, so your neutrophils, eosinophils, and basophils. So you know the percentage of what they should be. And remember, if your neutrophils are elevated, it's generally a bacterial infection, right? So those are the largest number. I usually use 60, 40, 830 is my count I give you, and I realize it comes to more than 100, but neutrophils should be anywhere between 50 to 60%. 90% if you have a bacterial infection or something's going on. Eosinophils are super low. Basophils are very, very low. All right, and as a keynote right here, basophils, their cousin is a mast cell, right? So basophils, when they leave the bloodstream, they become mast cells. Now, mast cells are in your connective tissue, all right, and these are a dendritic cell, partially, and mast cells are in your any part of your body that's exposed to the environment. So anything that has a mucous membrane is going to have a mast cell. So they have exact same responses to basophils. So there's going to be elevated if you have an allergic reaction. Basophils and mast cells are the same type of cell. They're very related. Basophils, all right, they're granules. They stay in the blood. Mast cells are their cousin. They're outside of the blood. Everyone got that? So here is your complete blood count, the different types of cells. So neutrophils, eosinophils, basophils. Monocytes are those huge cells. When monocytes leave your bloodstream, they turn into macrophages, it says right here. Right? Macrophages and dendritic cells. Now, macrophages and dendritic cells, I want you guys to remember this. They are antigen-presenting cells. So they literally go in, they consume anything foreign, they process it, and they literally present it to your immune system. So your immune system will make antibodies to it. Right? So antibodies, we'll talk about it later, but they kind of super tag things and they can jam up all the receptors. So if, I know I'm getting long-winded, but let's say that you, unfortunately, let's say you were exposed to COVID-19, all right? If you were able to produce antibodies to it and you had enough, if your body was working, if your immune system was up and running, your body could literally take the antibodies to COVID-19. It could actually put an antibody on every single one of those spikes. Now that virus, it got exposed to a type 2 pneumocyte, it could attach to your cell because every single spike is jammed up. I took my dryer. Do you guys know what a dryer ball is? can't see you, any guys. All right, but a dryer ball has those things you put in your dryer and they bounce around and get your clothes to dry faster. They have all those spikes on it. That's what a virus looks like. These macrophage and dendritic cells will present that to the virus, the components of it, to your immune system. It'll make antibodies. The antibodies will literally attach to all of those spikes. Now that virus can't attach to anything. Your erythrocytes, I'm hoping that you guys know erythro means red, cyte means big. So these are your red blood cells. They develop in your bone marrow. Stem cells, they lose their nucleus. So remember, they'll try to trick you. Please remember, your red blood cells always had a nucleus. All right. When they were erythroblasts, they had a nucleus. Your red blood cells eject the nucleus, so there's more room for hemoglobin. Your platelets are formed elements circulating blood. They're not whole blood cells. And I don't know if they told you in A&P, what platelets are made out of megakaryoblast, right? So megakaryoblast, they kind of get into your capillary bed, and as the blood comes through, it starts cleaving off parts of that, and that's what turns into your platelets. These are just part of megakaryoblast, and platelets are what we want. So when they leave the capillary bed or they're exposed to something outside the capillary bed, they're designed to clot, to stop bleeding. Your lymphatic system, all right, it has two functions. It has an immune function and it has a digestive function. I know I talked about this in class earlier. So your lymphatic system, the digestive function is to get any kind of lipids, small lipids into your digestive system, because remember, lipids and water don't mix. So lipids, they have to be, if they're really tiny, the lymphatic system could take them to your liver for processing, or we can coat them with chylomicrons of protein to get them to float around your bloodstream. But for today, remember, your lymphatic system provides an auxiliary route for return of extracellular fluid to the circulatory system. And we want that to happen because anything that's outside of your capillary bed can have potential pathogens or something foreign. And we want this to go to your lymphatic vessels where your B cells and T cells are, so that your immune system is aware of it and can start producing antibodies to it. Right? Acts as a drain-off system for the inflammatory response. When I talked about the reticular endothelial system before, I said as fluid leaves your capillary bed, as their pressure builds, if it gets super high, those valves in your lymphatic system will open up. That fluid goes in your lymphatic system and goes back to your immune system. So if you have an inflammatory response, more fluid is going to leave your capillary bed. That interstitial fluid gets bigger, the volume gets bigger, and that's what opens up your lymphatic system, and all that fluid drains into your lymph nodes, all right, to process any antigens and get that extra fluid back into your circulatory system to get reprocessed. All right, and once again, it renders surveillance, recognition, and protection against foreign material. That's its whole purpose. Right. Your lymphatic fluid is lymph-like fluid. Remember, this fluid comes from your capillary bed. So it's the same fluid basically as the fluid we saw when we looked at that test tube of blood, that serum. Right? Formed when blood components move out of the blood vessels into extracellular spaces, made up of water, dissolved salts, and 2 to 5% of protein. All right. And remember, you have lymphatic fluid or you have inflammation, your capillary beds are opening up. This fluid's leaving your capillary bed to go into your interstitial fluid. It's doing that because let's say you have a cut or a bee sting or a spider bite or whatever. We want to dilute whatever that toxin is, all right? And if you have injury, we want to get fluid, proteins, and any kind of white blood cells out of the capillary bed into that fluid to fight the infection, right? Remember, there's no Uber, there's no trucks to bring this stuff there. So we have to have a chemotaxis. We send out chemical signals to damage cells or whatever. We send out a chemical signal. Capillary beds open up. This fluid leaves your capillary bed with these proteins and your monocytes and all the inflammatory fluid to start cleaning up that mess. Right? transports white blood cells, fat, cellular debris, and infectious agents, all right, into your lymph nodes and back to your heart, all right, and your circulatory system. We clean it up, we filter it, and recirculate that fluid. So here's your lymphatic vessels. So just realize, I'm sure you went over this A and P, but remember you have your vessels. You have your vessels here, go from your arteries, arterioles, capillary bed, venules, and veins. But remember, you have lymphatic vessels all around in there. So when the pressure gets higher, this fluid, extracellular fluid, gets into your lymph fluid. It travels to your lymph nodes. B cells and T cells are here. It kind of filters it, wages in a war against it, brings it back into your right subclavian, or your thoracic duct, or your right subclavian back here. Right? Remember, it's a one-way direction. So if any of you have ever had a professional massage, all right, what they're going to do is they're going to go distal portion of the body. They always bring it towards your heart. So when they're doing that massage, they're actually taking all those lymph nodes, they're squeezing them all out. They're getting all that fluid, they're getting it into your lymph nodes, all right? And if you've had a massage done before, they tell you after the massage, make sure you drink plenty of fluids. We want to be able to flush all this out. So after a massage, you're getting all the stuff out of your lymph nodes. It's recirculating, getting all those toxins out of there, recirculating, flushing out the system. Here's just showing you the circulation of the lymphatic vessels. And just remember, the fluid is getting is higher out here. It's getting your lymph nodes. There's your afferent. So that's all coming like a traffic jam coming in here. It's percolating around your lymph nodes, coming back out here. And then once again, it goes to the lymphatic ducts back to your heart. The time check, am I running? Okay. All right, so your lymph organs and tissue. Remember, your primary lymph node organs are your sites of lymphocyte origin and maturation, so your thymus and bone marrow. Anybody remember the function of your thymus? All right, this is where your T cells go to learn. They have to either declare they're going to be a CD4 cell, a CD8 cell, a killer T cell, or they have to become, CD4 or CD8. So they have to decide it'd be a CD4 or CD8, all right, and they have to be able to tell self from non-self. Right, so if you're in the lymph node, if you're in the thymus and you don't know self from non-self, your body will do something called clonal deletion. It'll take that T cell, and it'll kill it because it wasn't able to tell self from non-self. The problem is if a T cell gets past that and it can't decide self from non-self and it gets away and we don't delete that clone, it starts reproducing and now we have autoimmune diseases, right? Because that T cell, we have a huge problem with the thymus and other organs that they go in and start attacking your own organs because that T cell didn't pass its test in the thymus. We have our secondary lymph organs, all right? So does anybody remember Peyer's patches or tonsils? All right, so these are secondary lymphoid organs and tissues, circulatory-based locations such as spleen and lymph nodes, collections of cells distributed to the body, tissues. These are in your skin, and then once again, your mucous membrane. So all of these, I'm sure you guys still don't believe me, these are all outside your body, all right? So we want, gall is GI tract, this is your membranes, all right, Peyer's patches in your digestive tract. So we want, if there's any kind of pathogen that made it into your mucous membranes, your digestive tract, we want it to get in here. So you can think of these as little lymph nodes. You can think of them as, you guys know what those fly strips are? Those really sticky fly strips they have that catch flies, the nasty things. No, we can think of it like that, or your tonsils. Any of these foreign invaders come through, they get into any of these mucus associated membranes. We can send the dendritic cells will capture them, send them back to your lymph nodes, and we can have an immune response immediately. So they're on any part of the body that's outside of the outside of your body. And so your thymus is a high rate of growth and activity until puberty, begins to shrink after you're about age 20. All right. When you be my age, you can barely find it. All right. So the reason is we have a theory that you're going to be exposed to most of your pathogens before the age of 20. All right. So you want your thymus there, high rate of growth and activity into puberty. After that, you're going to be exposed to pretty much everything. You don't really need your thymus to be that active. All right. There's other theories that say if you pound on it or rub it, it'll start growing. And if you look at a lot of primates before they go into battle, they start pounding their chest. They're stimulating their thymus because they realize they may get caught or they may be injured. They want to stimulate that thymus in case they come across foreign particles. Your lymph nodes are small, encapsulated bean-shaped organs. We saw that before. Remember, these are all through your body, and they have a lot of afferent channels, one efferent. We want that. We want all the fluid coming in. We want to slow it down. We want to have a chance to percolate around and process. Your spleen, structurally similar to lymph nodes, filters circulating blood to remove worn-out red blood cells, pathogens. All right, A and P, they should have told you your spleen has a red portion and a white portion. All right, your red portion, I know you guys are really smart. It's for to get rid of your worn-out red blood cells. All right, because remember, these red blood cells are the size so small they can only circulate through your capillary bed, and they're biconcave. They can bend and twist. But after they're supposed to be reformulated every 120 days because remember, they don't have a nucleus. They can't regenerate. They automatically, your spleen, as the red blood cells pull up, the spleen's like, dude, you're beat up. You're all ragged. We're going to rip you apart. We're going to take your heme. We circulate that iron. We're going to take your globulin, that protein. We're going to reuse that. And your spleen will remake your red blood cells or get rid of all your old red blood cells. And the white portion is for all of your white blood cells in that portion of the spleen. Right? Miscellaneous is your gall, your associated lymphatic tissue, and Peyer's patches. Once again, these are outside of your body, and these are going to be small lymph nodes for anything circulating in that area to pick up anything foreign and present these cells to your immune system. So your action of your second line of defense, we want recognition. We want to be able to recognize something as foreign. Right? Once again, we might not know exactly what it is, but with those pathogen-associated molecular patterns, we kind of know it's not normal. We don't know what it is, but we're going to phagocytose it and present whatever those HLA foreign markers are on that cell or that bacteria, present it to your immune system and say, listen, I don't know what this is, not my job, but here, I found this. Figure out what it is, make antibodies to it, and we want to process that. Inflammation, we want inflammation. If there's any kind of damage or something foreign, we want to present inflammation. We want fluid to leave your capillary bed. We want your white blood cells to leave your capillary bed. We want to increase blood flow to the area. Why would we want to increase heat? Why would we want to elevate your temperature? Anyone have any idea? Remember, a lot of these things only have a small degree of temperature they can live in. We elevate your temperature, we spike a fever with inflammation. All right, a lot of it can't grow in that temperature range. And if we increase your body temperature, it increases your metabolic rate, increases the activity of your immune system. Your immune system says, oh, wait a minute, I have a fever. You guys wake up. We got to get busy. Something's going on. We want to phagocytose anything foreign. We want to engulf it. We want to present it to the immune system. Interferon, I said before, is a chemical that your immune system sends out. Your natural killer cells send out. Their virus, anything that's been infected with a virus, we send out that chemical. And we tell any cell or virus or cell in the area, if you've been infected with a virus, I want you to go through apoptosis. We don't want that cell to be reproducing viruses. The complement system we'll get to at the end. Complement system, we can actually neutralize certain things, or we can opsonize them. We can actually go in and tag them. For the immune system, we say, listen, this is foreign. The immune system, as it comes by, it can't miss it. It's kind of like putting an LED blinking light on something. Your immune system can't miss that. Or if we do neutralization, I said before, if we can go in and put antibodies on every single spike or receptor on a virus, even if it lands on type 2 pneumocyte, or if it's hepatitis virus, if it lands on your liver cells, it can't get in because the receptor lock and key is jammed up. It's kind of like, you didn't hear it from me, but if you were pissed at someone and you were to put super glue in their lock, they can't get in, right? You didn't hear that from me, but that's kind of what we do. We can go in and we can super glue the lock so nothing can get in. Right, so inflammatory response, I'm assuming this is all review for you. Redness, we increase circulation, vasodilation. We want to, if there's something going on, we want to get more blood to the area. You've been bit by something, we want to be able to thin it out. If it's a toxin, we want to be able to thin it out. Warmth is given off by increased blood flow. Your swelling, all that fluid is leaving your capillary bed into that endothelial reticular system, and your extracellular fluid. We increase that pressure. Once again, it's going to open up those lymph vessels, and we want that. We want the lymph fluid. We want all that extracellular fluid to go to the lymph system, because we're assuming if you damage that, if bacteria got in, a virus got in, some kind of foreign rickettsia got in, we want to bring that to your lymph system. We can filter it out before it actually gets into your systemic circulation. We want that. And then pain and stimulation of nerve endings. Remember, only your nerve endings can actually feel pain. Nothing else in your body can feel pain. So if you're feeling pain, something is pissing off your afferent dendrites, right? Something's causing it. So it's inflammation, pressure on it is causing pain. Your body, it's stimulating your body to do something about it. Remember, your thymus edits out 90% of all your pain messages. So by the time you feel pain, there's a lot of signals going up to your brain. Your thymus is cutting out 90% of it. So if you actually feel pain, it's serious. Your body's saying, listen, there might be 1 million signals going up, but only 10,000 got through. Right? So if 10,000 got through, your brain's saying, listen, your body's saying, listen, there's something serious going on here. So this should all be reviewed. Here's the major events of inflammation. We have damage. Let's say these are bacteria. We want to increase the fluid in the capillary beds in there. We want to increase that fluid, get that pressure up. We want it to get into your lymphatic system, into your lymph nodes, so we can filter it out and hopefully wage an immune response to it. Here's some chemical mediators of the inflammatory system. All right, so we've all probably heard of some of these before. Histamine, serotonin, bradykinin, prostaglandins. These are chemical mediators with vasoactive effects. So these are going to cause your blood vessels to vasodilate. All right, and if anyone's had an allergic reaction, you're going to take antihistamines. All right, because if you have all these chemical mediators going on, it's going to cause you to itch. Your mast cells are going to go crazy. Bradykinin and prostaglandins are pain-producing chemicals. And anyone who's taken Advil or Tylenol, if you've ever heard of these, it actually breaks down prostaglandin and bradykinin. It breaks down that whole process, so you don't feel pain. That's how it works. Right? Mediators both cause vasodilation and chemotactic effects. Remember, chemotaxis, all right? Chemo is chemical, taxis is movement. So once again, your body doesn't have Wi-Fi. It can't text. It can't call each other up. There's no cell phone reception there. So the only way it can get things to come is it sends out chemical messengers, all right? And your body will follow these chemical gradients, all right? It goes from low to high. As it will pick up the smallest amount, it'll just follow it towards the greater concentration. That's how these cells find their way. We're sending out chemical mediators. Remember, if you're if you damage a cell, all right, so the cytoplasm of that cell actually it breaks open. The fluid inside that cell is out in your extracellular fluid. It doesn't belong there. So you're changing the chemical concentration of that, causing your red blood cells, or your white blood cells, and these monocytes, they're going to say, wait a minute, that doesn't belong there. I'm going to go investigate. And it's going to start following the chemical concentration, or, call it blood crumb trail or whatever. It's going to follow that towards that area and say, wait a minute, the concentration is getting bigger and bigger, more and more. I'm going to go towards that area. They find the problem and they'll do whatever they can about it. So diapoesis, I said before, PED is foot. All right, so this is when your capillary bed opens up, and if you see, these neutrophils are following this chemical gradient. So let's just say that we had injury here. We had a cell that lysed or broke open, or there's inflammation or a toxin there. These cells actually are they're finding this chemical concentration. They're going to follow it wherever the problem is. So chemotaxis is migration in response to specific chemicals at the site of injury or infection. All right, and I said before, the benefits of fever. We want to increase interleukin 1. And remember what interleukin does? Remember I said it's going to cause these cells that are infected to kill themselves so they don't produce more viruses. So pyrogens are things that cause your hypothalamus to increase your body temperature. All right, and once again, your body temperature, you guys can do whatever you want, but today when I went to go to ECC, they literally took my temperature, 97.5. I guess I'm going to live. All right, but they go in, your body increases its temperature because, once again, a lot of these pathogens can't live in that temperature. They're not going to find you hospitable. They're going to leave. They're going to die. All right. And it increases your metabolic rate, causes your immune system to ramp up its replication, and it ramps up its efforts. All right. So benefits of fever: inhibits multiplication of temperature-sensitive microorganisms, impedes nutrition of bacteria by reducing the available iron. Remember I said last week, your body's going to sequester iron and zinc, all right? But if you're staph, you have a virulence factor, you can produce hemolysin. The bacteria will go in, it'll secrete a chemical that will lyse these red blood cells and grab all the heme at once. All right, so that's a virulence factor. Increases metabolism and stimulates immune reactions and protective physiological processes. Like I said, it's going to ramp up the activity. So you can do whatever you want. Like I said before, if you want to take Tylenol, if you have a temperature of 100 or 99, that's fine. All right, but there's a lot of homeopathic doctors and a lot of people right now are saying, you know what? Let the fever elevate. We want all these things going on. If the temperature becomes damaging to the point where it's going to denature our proteins, specifically our enzymes, then we want to do something about it. But until then, let it raise. It's a little bit uncomfortable, but we want these benefits of that fever. If you take anything pyrogenic medication that's going to drop that temperature or release that fever, you're not going to get these benefits of the activity. Phagocytosis, once again, these are going to be sites or grown cells are going to go in, they're going to consume this bacteria or something foreign. Like I said, they won't know exactly what it is, but they realize it's not quite normal. They automatically know it's not normal. They go in and consume it. To survey tissue compartments and discover microbes, particulate matter, and dead or injured cells. They go on, they figure out what it is. And remember, we also, before I forget, when we're talking about autoimmune cells and natural killer cells, we want some of our cells to go in and kill our own cells if they're injured or damaged, all right? We also want some of our CD4 helper cells, they go in and they kind of tell the immune system, calm down, you're overreacting. We'll talk about that too. I want to make sure I mention that in case I ever forget. All right, they ingest and eliminate these materials. They extract immunogenic information from the foreign material. So when we get into this later on, remember these dendritic cells, These antigen presenting cells, they go in, they engulf it, they process it, and like I said, they wrap it up nice and pretty, and they literally present this to your CD4 cells. And your CD4 cells are your helper T cells. They talk to your B cells and T cells, and they all kind of agree, listen, this is foreign. We're going to agree it's foreign. We're going to shake hands. We're going to sign a contract. And from that point on, we're going to make antibodies to it. And then the T cells will go in. If they see that antigen in any cell, they literally go in. Your T cells have hand-to-hand combat. They go in, attach to that cell, and they poke a hole in it. They shoot some toxins into it, and they cause that cell to lyse and die. We'll get into that later. I just want to bring it up so you hear it 4 or 5 times before the end of the semester. So at the beginning of the semester, I know I went through the CBC with differential, but here, if I ask you guys a question on your exam, remember I said neutrophils should be 50 to 90% of your white blood cells. If they're elevated, generally it is a bacterial infection. General purpose, react early to bacterial and other foreign materials and to damage tissue. The other thing you should know, these can technically be antigen presenting cells, and these are always the first responders. These are the EMTs. These are always first responders to the scene. Eosinophils, generally when you see this, it's always a pathogenic infection. So it's a large parasite, right? So generally worms or something going on, and antigen-antibody reactions, and sometimes it can be in allergies along with your basophils and mast cells. But generally, if I ordered a CBC with differential and that comes back and I see eosinophils are elevated, I'm going to ask the patient, listen, guys, were you somewhere foreign? Did you eat sushi from gas station? You know, looking for Chinese liver flukes or some kind of worm infection. So eosinophils are parasitic infections of some sort, tapeworms or liver flukes or something weird. All right. Macrophages or macrophages. All right, macro means big, phage to eat. So these are big eaters. So these things have a voracious appetite. They're derived from monocytes. Remember I said, monocytes, when they leave the bloodstream, become macrophages. Scavenger and process foreign substances to prepare them for reactions with B and T lymphocytes. So these, once again, are antigen-presenting cells. So their job is to go in, engulf, process, and present. Right, you guys got that? And macrophages and dendritic cells. All right, macrophages are free circulating. Dendritic cells, and we talk about these later. All right, these are not dendrites like in your nerve endings. Dendritic cells are in your tissue. All right, so they're in your tissue, your connective tissue, or your mucous membranes, or anything on the outside of the body. So these are going to go in and they're going to present foreign material that's on the outside of your body somewhere in one of your mucous membranes. So recognition of foreign cells, right? So we'll get into this a little bit in the next semester or the next chapter. Protein receptors within cell membranes of macrophages are called toll-like receptors. Remember, macrophages were your antigen-presenting cells. So they go in, and these are like antennae, or if they were, let's just theoretically say they were blind because they can't see. They go and feel like Braille. They're going to feel what's going on, and they're going to look at other cells. And if something seems foreign, doesn't seem right, they go in and engulf it. They can't see what it is. They just know it's not right. And macrophages, once again, macro means big, phage means eater. These things will go in and they'll attack anything. They don't care what size it is. They attack foreign molecules and signal the macrophage to produce chemicals to stimulate an immune response. All right, so the mechanism, chemotaxis, remember this is chemical taxis. These are the chemicals that are given out or that are foreign to the body. These cells are going to follow this chemical gradient. They go from low concentration to high concentration. They follow the chemical gradient. All right, they ingest it. Remember, they recognize pathogen-associated molecular patterns. They don't know exactly what it is, but they know it's foreign. These are not super specific. These are just general. This is second line of defense. And phagosome, all right? So phagosomes are these chemicals that these cells are going to give off. It could be lysosome, it could be an enzyme, it could be peroxide. It's some kind of chemical that's going to go in and start dissolving or eating these cells. Right? And these can cause death within 30 minutes if it can get inside the cell. All right. And remember, your T cells, I said before, they're going to have hand-to-hand combat. They're going to actually they're going to put something called perforins into cells. They actually poke a hole in it, and they literally vomit this chemical into the cell and cause it to be destroyed within 20, 30 minutes. Right? Destruction and elimination. Right. And once again, I said before in the beginning, oxygen-dependent system, respiratory bursts. So these cells will actually go in and they will inundate the bacteria with oxygen. Right? Liberation of lactic acid, right? Lysosomes and nitric oxide. So these are chemicals that are very, very toxic, or can go in and give it peroxide or superoxygen dismutase. They'll go in and give it this oxygen, and the cells can't deal with that much oxygen. Remember, I said before, beginning of the semester, oxygen is very toxic to us. Everything is toxic, but if we can process it, it's fine. When you can't. So if you're a bacteria and you can't process oxygen, you become anaerobic. Remember me talking about that? So here is phagocytosis. All right. So it's going to go in, engulf this. It starts processing it, right? Processing the chemicals. It's going to start taking off all of these receptors, all of these HLA markers on the bacteria, goes in and takes these substances and it processes them for the immune system. This is not going to play, but I think this I embedded in Blackboard for you guys too. Right. Sorry, we're playing my MacBook Pro. I don't know what's going on, but it just won't do it. Right. And once again, this is that interferon. All right, you guys don't have to know interferon alpha, beta, and gamma. All right, but these are small proteins produced by certain white blood cells and tissue cells, produced in response to viruses. All right, RNA, because most of the viruses now are ribonucleic acid, not DNA. The scary thing is they can go right into a cell and start replicating. They don't have to go back. They don't even have to go into the nucleus like DNA. We talked about transition and translation before. These can actually start processing right in the cytoplasm. They can start replicating in the cytoplasm. They go right to the nucleus, start regenerating, and send out viruses. Immune products and various antigens, they bind to the cell surfaces and induce expression of antiviral proteins, inhibit expression of cancer genes. They go in, they bind to it, and they'll literally tell the cell, listen, you come across this, don't let it in, and if it's inside, I want you to produce apoptosis. And complement, we're not going to get into the big details on this, but I do want you guys to know what neutralization and opsonization is. All right, neutralization, we go in and we can kind of jam up the receptors for it, neutralize it more or less. All right, and opsonization, we can go in and we kind of super tag the pathogen. We kind of put little LED blinking lights on it and kind of let the immune system, they can't miss it. All right, guys, let me do a time check. All right, everyone's okay for another hour? Well, anyway, if you're not, that's fine. I'm going to go switch over the PowerPoint. At least we'll get into part of chapter 15. All right, so give me a couple minutes here. Grab a drink, take a smoke break, whatever you need to do. I'm kidding about the smoke break. Brianna, how are you? Can you hear me? Can you hear me?
Yes,
Okay. Thanks for your help last week, by the way.
you're welcome.
Let me find this PowerPoint. Good Lord. It's going to be the death of me. That's patho either. I still want that. No. Seriously. All right.
I like working from.
You guys realize I'm totally incompetent on this computer, right?
So it's okay.
Huh?
For all of us, so it's okay.
Oh God, I just, you know, I think I spent 2,400 hours on this computer and I can't even get it to work. It's ridiculous. So it should be here. All right, in chapter 14, I loaded 2 versions of it. If it doesn't, one of them doesn't open the other version of it. I don't know. I had to go way back in and pick out an old version because it just would not work. Enable that. Okay. All right. All right. Everybody's okay, or should I give them another minute? I only see Garrett on here. Anybody else here? Hello? Bueller? Anyone? All right, I'm gonna go grab some ice, and then hopefully the rest of us will join. All right.
It's funny because Garrett is put up as Phil Jones.
I'm put up as Phil Jones.
You're up as the instructor right now, yeah.
I found a button that will let me take the host position from him.
I know, me
Well,
too.
she must have done, yeah, I was gonna say, because that's what Garrett did too.
Okay, so who's host right now?
Well, now it's Katrina, or Katarina, sorry.
Oh, I never clicked anything. He's still listed as host for me.
Still
Oh,
here?
that's so funny.
You're still here, don't worry.
I just don't see you.
All right. All right, let's spend a little time on this. We probably won't get through all of it, but we'll get through whatever we can here. All right, so I want to get into specific immunity. All right, so this is going to be a little bit more difficult. So I don't know if they went over B cells and T cells with you at A&P, all right, but this gets a little trickier. All right, so this is adaptive line of defense. So please realize you guys are not born with this. So this you have to be exposed to, and you have to produce specific antibodies to it, right? And your body's going to produce antibodies to an antigen, right? So you guys remember I said at the beginning of the semester, you can't wear patchouli, right?
Mhm.
All right, because it's an antigen that I am highly allergic to, right? So adaptive line of defense, or cat dander, right? I am allergic to that. Or it's a hapten. It's very, very tiny, right? I shouldn't be allergic to it. It's a normal thing, all right? But I have a hypersensitive reaction to. Dawn, I love it. Wine, awesome. Yes. All right. Is that a Pinot? Can't hear you, but I'm assuming. Okay.
It's a Cabernet.
Huh?
It's a Cabernet.
Okay, good. It's nice and dry.
Mhm. Very good.
Nice.
Mhm.
All right, so your third line of defense is acquired. So this is dual system of B and T lymphocytes. All right, immunocompetence. All right, so here's the thing. What would happen if you guys weren't able to produce antibodies?
We need to look
Very
up
bad thing. All right, so we can't antibodies. We actually go in and we tag the antigen antibody. We kind of make a great big, take the antibody and we attach it with the antigen, and we attach an antibody to it. We have a great big complex, all right? So your immune system can't miss that. It's huge, all right? If you can't make antibodies, you're going to have missing piece of it, all right? Or you could have something called the Georgia syndrome, where you don't have a thymus. So if you don't have a thymus, now you can't make CD4 cells or CD8 cells, all right? And remember I said before, without a CD4 cell, it literally triages your entire immune system. So you could literally have, if you don't have a CD4 cell, you could have B cells and T8 cells out there. They're standing around, they have no idea what to do. They're just standing there waiting to be told what to do. That's why AIDS is so deadly, because your CD4 cells are so diminished that your immune system has no one telling it what to do. It'd be like having an emergency room with no one triaging it. So your surgeons, everyone's just standing there waiting. No one's being told what to do. It's devastating. All right, so an antigen is a molecule that stimulates a response by T and B cells. Two features that characterize specific immunity. All right, these are crucial. Specificity. Antibodies are produced, function only against the antigen they were produced to respond to. All right. Remember, antibodies are produced by plasma cells, which are derived from B cells. All right. So once again, I said they're going to try to trick you on your NCLEX or your board exam. They're going to give you antibodies. You're going to have a B cell and plasma cell, and you're going to automatically want to say, oh, B cell. All right. That's true if plasma cell's not there. But if plasma cell's there, plasma cell is the answer. Right? Plasma cell's not there, obviously it's B cell. Right? But plasma cells are derived from a B cell. Memory. Lymphocytes are programmed to recall their first encounter with an antigen and respond rapidly to subsequent encounters. That is the entire premise behind vaccination. With vaccination, we want to induce an antigen into your body. We want your B cells, all right, to produce plasma cells to produce the antibody, and we also want it to make memory cells. We want it to make memory cells so that if you get exposed to that antigen again, the memory cell already has the recipe in the card catalog. It pulls up the recipe and says, "Oh yes, we were exposed to this before. I can." It can take 5 days to 10 days to make the antibody. But if you have prior exposure, you have memory to it, we can produce that within 12 to 24 hours, which is a much faster response. And now they're saying, I keep bringing up COVID-19, but they're saying that people may have been exposed to this as early as November and December, and they have antibodies to it. So if we're exposed to it again, our body says, oh, wait, we've seen this before. We'll produce antibodies to it, and they're not getting sick. All right, overview of specific immune responses. Separate but related activities of the specific immune response. Development and differentiation of immune system. We want to be able to develop it, and we want to be able to differentiate between self and non-self. Lymphocytes and antigen processing. So lymphocytes are your white blood cells, your adult white blood cells. Antigen processing, I said before, were done by your neutrophils. Can do it. It's not their primary job, but they can do it. Macrophages, all right, and monocytes, when they leave the bloodstream, return to macrophages. Macrophages can do it. And remember those dendritic cells that are in your outside of your body, in your tissues, and in your mucous membranes are the dendritic dendritic cells, or antigen presenting cells. The cooperation between lymphocytes during antigen presentation. Once again, these macrophages and dendritic cells, they engulf something, they digest it, and they process it, and they present it to your immune system. All right, B lymphocytes in the production and actions of antibodies. Antibodies, like I said before, these are antibodies we attach to antigens so the immune system can't miss it. And then your T lymphocyte responses. So we have your B cells, right? Your bone marrow sites are where all of your bone marrow cells are produced. So your white blood cells, red blood cells, your plasma cells. And remember, all of these processing of cells is all generated by endocrine processing in the kidney. Kidney is going to produce erythropoietin and thrombopoietin and all these poietins that are going to tell the bone cells to produce more of whatever specific cell it is. And then your T cell line of defense, right? Your thymus, right, is going to tell your body what T cells have to decide whether they want to be CD4 cells or CD8 cells. They have to decide what they want to do, and they have to remember they have to be able to tell self from non-self. Right? We'll go through all this as we go along here. But here's kind of a chart of what's going on. All right, so development of the immune response system. Cells, receptors, and markers convey specific identity of the cell. Major functions of the receptors. So these are the receptors on these macrophages, dendritic cells, and some neutrophils. They have to perceive and attach to non-self and foreign molecules. They have to, those toll-like receptors have to be able to tell what it is. They have to attach to it. They have to recognize that if it's self, they let go. They say, okay, never mind. They have to receive and transmit chemical messages among other cells of the system. They aid in cellular development. All right, so these cells, if they see it's foreign, they attach to it and process it. If they recognize something to be self, they leave it alone. All right. The problem is, what if you're a cancer cell, all right, or a virus-infected cell? A lot of times, the markers on the outside of your cell are going to look completely normal. All right, so your immune system, it's going by, and these toll-like receptors are looking at the cell, and everything looks normal. If it's a cancer cell, and it starts changing the HLA type 1 receptor in the cell, if it changes those, the immune system will say, wait, it doesn't look right, they'll attack it, right? Or if it's a virus-infected cell, if it's not infected badly, or if it hasn't started changing its cell surface markers, the immune system thinks it's completely normal, right? And I said before that we have some of these bacteria that they trick macrophages into engulfing them. All right, like Listeria and Shigella and Salmonella, they taunt the macrophage into engulfing them, and they start reproducing inside the macrophage. And the macrophage can go anywhere it wants. The immune system thinks it's completely normal. It is completely normal, and these bacteria and these other things can start growing inside these macrophages, and they elude the entire immune system. So we're going to do major histocompatibility complex, MHC, MHC1 markers and MHC2 markers. So here's the thing. Major histo tissue compatibility complex. All right, so if you, HMAC1 and HLA, human lymphocytic antigen, are the same thing. It's a different word for the same thing. These are the markers on your cells, and unless you have an identical twin, all right, every single person on the planet has different HLA or MHC markers on their cells, which is completely amazing. We have the amount of population we have, we realize all of them are completely different unless you have an identical twin. Right. The other thing is, if you ever heard about organ transplants or like a bone marrow transplant, we go in and we look at all of your HLA or MHC markers, and we try to find a match that has the most these that are compatible. All right, I don't know what the numbers are anymore. It could be, I think it's like 9 out of 12 or 7 out of 12 or whatever is the bare minimum. So if these markers are very similar, we can go and do the bone marrow transplant or the organ transplant, because as the new host, as you start looking at these cells, they're very similar to your own cells when you go to do the organ transplant. And the immune system won't attack it because these markers look very similar to yours, and you won't have as much tissue rejection. All right, also known as human leukocytic antigen, HLA or MHC. Right. MHC1 are the same as HLA markers. All right, we're going to be talking about MHC2 markers. All right, so when you see MHC2, when you think of two hands, they're going to be presenting MHC2 markers, present a foreign antigen to the immune system. All right, so MHC2 are presenter cells. All of the dendritic antigen presenting cells have MHC2 markers, and they present the antigen to the immune system. And so, listen, I captured this, I processed it, Now, your job is to go and produce antibodies to it. Plays a role in recognition of self by the immune system and in rejection of foreign tissues. All right. So this is a class 1 MHC molecule found on all nucleated human cells. All right. The only problem with that statement right there is some books will say that red blood cells don't have them. Other books will say they do have it. So that's a gray area right now. All right. So this MHC1 marker is the same as HLA marker. Now, class 2 MHC markers are found in some types of white blood cells. These are your antigen presenting cells. And remember, the easiest way to memorize this looks like two hands. They're presenting something. Right? You guys got that? Maybe, yes. All right. Genes for MHC clustered is a multi-genome complex. All right. And like I said before, unless you have a twin, they're all going to be different. Class 1 markers display unique characteristics of self molecules and regulate immune reactions required for T lymphocytes. Remember, your T lymphocytes are your CD4 and CD8. Class 2 regulatory receptors found on macrophages, dendritic cells, and B cells. These are involved in presenting antigens to T cells. So class 2, once again, these are presenting cells. Lymphocytes are your adult white blood cells. Lymphocytes' role in surveillance and recognition is a function of their receptors. That's how they tell. They said they don't have facial recognition cameras. They don't have Wi-Fi or whatever. Right? They just have those markers on the cell. And they go in and those toll-like receptors, they go in and like Braille, they kind of figure out what's going on and they check for chemical changes. All right, B cell receptors bind free antigens. These antigens, again, these are those foreign materials. They could be haptens. They could be anything foreign. Antigenic or antigens are something that you're going to produce antibodies to. T cell receptors bind process antigens together with the MHC molecules on cells that present antigens to them. All right, so T cell receptors. After these type 2 cells process and present this, the T cell bind to these antigens together with the MHC complex in the cells, and they present them to antigens. The antigens are presented to the T cell. The T cell says, yeah, you know what? You're right. That doesn't look right. They agree it's foreign. They agree, they handshake, and then this arm comes along and attaches the two of them together, and they literally sign a contract. All right. Once the arm comes along and attaches to it, it's locked and loaded, and now it's a death sentence for anything that has these receptors on it. That T cell, T8 cell, if it sees that marker on a cell, it goes in and destroys it. And that's a problem with these people that have these immune deficiencies, because the antigen presenting cells will go in and they'll take part of your cell, of whatever organ it is, whatever you have an autoimmune disease against, it'll present it to the T cell. T cells, okay, that looks foreign, you're right. They agree. Now that T cell, every time it sees that marker on any organ or tissue, it goes in and literally destroys it. All right, clonal selection theory. So lymphocytes use 500 genes to produce a tremendous variety of specific receptors. All right, this is where it gets amazing, guys. All right, so please realize your lymphocytes, we have 500 different gene combinations. So we literally do a quick pick. These B cells go in, they just start doing a quick pick. They take certain sections of each part of your gene, they mix them all together. They make all these different combinations. And these B cells just sit in your lymph nodes for the rest of their life, waiting for something that looks just like that. So just imagine you had 100 million different lottery tickets, and they're just sitting in there waiting for you to pick that specific sequence so you win the $10 million. That's what these B cells do. They sit their whole life waiting to come across an antigen. Undifferentiated lymphocytes undergo a continuous series of divisions and genetic changes that generate millions of different cell types. Right, it's more like 100 million. Right? Each cell has a particular unique receptor specificity. And once again, these B cells are specific for a certain thing. They may never come across it, but we make all these crazy combinations, just waiting for the off chance that we come across this antigen. Right? And this is where some people have great immune systems, some people don't, because their B cells, they may not have that many different combinations, or they may not have generated the combination that is specific to that certain antigen. So these people get sick more often, or why are some people immune to some diseases and some aren't? It depends on their B cell. That B cell clonal selection made some combination that was able to produce an antibody where other people couldn't. So the development of your lymphocytes. Remember in A and P, they told you B cells, they're produced there, and the B cells, they come out fully integrated, fully knowing what they're doing. T cells, they have to go to the thymus and differentiate between CD4 and CD8, and they have to be able to tell self from non-self. So in the bone marrow, lymphocytic stem cells differentiate either into T cell or B cells. B cells stay in the bone marrow while T cells migrate to the thymus. That's why they're called T cells, because it's the thymus, because it runs in a T. Both T and B cells migrate to secondary lymph tissue. All right, these are your lymph nodes, your Peyer's patches, that gall, your tonsils. They're going there waiting to come across something foreign. Right? You guys remember what IG was? Immunoglobulin receptors. All right. No? Okay. So let me go back here. Oh, sorry. So lymphocyte development. Lymphocyte specificity is pre-programmed, existing in the genetic makeup before an antigen has ever entered the system. So once again, these lymphocytes, they go in, and I'll show you how it's done simplistically. They go in and they take pieces of each part of your genetic makeup and they shuffle it all around like a lottery. And they make up these pre-programmed genetic antigen receptors without even ever being exposed to it. So each genetically different type of lymphocyte is called a clone, expresses a single specificity for a specific antigen, whether it's seen it before or not. All right, and here's the thing. I mentioned clonal deletion before. So they're saying these are your lymphocyte stem cells. We're making all these, if you look, all these markers are different. Theoretically, there's hundreds of millions of them, all right? So they're saying if these lymphocytes, these are adult white blood cells, if their receptors find that the lock and key mechanism works for any of your self cells, they are supposed to go through apoptosis. It's called clonal deletion. The problem is these lymphocytes, your lymphocytes are your T cells or T cells. If they don't kill themselves, or they don't go through apoptosis, or if they cheat on their exams, all right, this is when these T cells, if they don't kill themselves, later on in life, they'll start going up to your thymus, or let's say your kidney or your endocardium or whatever, and they'll say, oh, you're foreign. I remember when I was a teenager in the thymus that my lock and key mechanism fit and I didn't kill myself, so I must be right. This is when they go in and they start doing autoimmune diseases, right? Because they didn't clonally delete themselves when they found your markers to be antigenic. So clonal selection, first introduction of each type of antigen into the immune system selects a genetically distinct lymphocyte. So again, each specific antigen, whether it's cat hair, dust mites, cockroach droppings, you can name anything that's an antigen. Could be pine needles, grass, pollen, whatever it is. Each one of these is very distinct for a very specific antigen, causes it to expand a clone of cells that can react to antigen. So the minute you're exposed to an antigen that you find foreign and your body makes antibodies to it, right? We make a bunch of clones. We say, you know what? We need a whole bunch, we need an army. We had one that recognized it as foreign, they agreed on it. Now we need an entire army, and we want memory cells. In case we come across it again, we can ramp up the army. So specific B cell receptors are immunoglobulins. IG, we talked about IG, we didn't, maybe it was in patho. IgA, IgE, IgM. Right, so you've heard of immunoglobulins before, or your titer, blood titer. We're checking blood titers for that. So receptors of genes of B cells govern immunoglobulin synthesis, IG. Immunoglobulins are large glycoproteins, so sugar proteins that serve as specific receptors for B cells. All right, here's the deal. T cells are hand-to-hand combat. B cells and plasma cells, remember, they make antibodies. Right? So immunoglobulins, these are the epicytes, or these are the epitopes where they bind onto the B cell. All right, there's a heavy chain and a light chain. This is the variable region. All right, so the heavy chain and all of them are the same. The only thing that changes on a B cell are these variable V regions. All right, and this is where the antigens actually bind. Right? So the other thing I want you guys to remember, this is a kind of like a Y here. So literally, this thing can actually change shape. It can go in and play twister. It can go in and bind. All right. And remember, a lot of these antigens have more than one. A lot of these foreign things have more than one antigen. So you could literally have 5 or 6 different B cells with different variable regions attaching, and they could be doing all kinds of contortions. And the cool thing is, if we had something foreign, if we could get 5 or 6 different B cells to attach to it, your immune system can't possibly miss that. Can't possibly miss it. Now, whether it's strong enough to attack it is one thing, but it can't miss it. Right, so specific B cell receptors or immunoglobulin are composed of 4 polypeptide chains. Right, so you have 2 heavy chains here and 2 light chains. Right, and once again, remember, the light chains are, these are the variable regions. This is where the antigen actually binds to it. So the Y-shaped arrangement ends of the fork formed by a light and heavy chains contain a wide range of variable antigen binding sites. These are where the antigens actually bind to it. All right, so there's a variable region. This C is your constant region, also known as the heavy region. All right, so the other thing, so we mentioned it again, this is what actually attaches to the cell membrane of the cell. All right. The other thing is, do you guys remember what is eosinophils do when we talk about the blood count? That was going to be for parasitic infections, or you had an allergic reaction. So let's say you had, this was floating around, your antibodies were floating around in your blood. Did you guys know IgA, IgE? So IgE is floating around your blood and you come across something you're allergic to. This IgE, this was an IgE antibody attached to something foreign, all right? This will go in and float around your blood. This part here will actually, I said it touches the cell membrane. If this attaches to a mast cell or basophil, right, it attaches there. Once it lands on that space pod, lands on that cell is going to start pumping out histamine, like 10,000 molecules of histamine per second. Right? That's how these work. It's amazing. But here's the development of the receptors, all right? So you guys don't have to know the specifics of this, but this is what's going to happen. Your body's going to go in, it's going to quick pick a bunch of these different genetic markers here. It's going to reshuffle them up, all right? And then remember, these are your variable regions. So these are just sitting on all your B cells. All these different combinations are sitting on your B cells. They're just waiting their whole life to have something float by that matches that lock and key. And then they jump up and down. They say, hallelujah, I've been waiting my whole life. They start making antibodies to it, right? They make a whole bunch of new antibodies, and then they make plasma cells, and they make memory cells. So if you ever come across it again, you automatically have the recipe. So these B cells are underneath, they're little virgin cells. They're just waiting for their first encounter with an antigen. Like I said, it may never happen. They're just waiting. They're in your lymph nodes, floating around, just waiting their whole life to have this quick pick lottery get drawn, and they can start acting. So there's just how this is how their development combination cause, we just recombinate. So all you guys that I care that you know about is these just recombine like a quick pick. You pick one of these, or 2 of these. They just kind of shuffle them all up, and they just make all kinds of B cells, and they're naive, just waiting for a specific antigen to come along. So these are your T cells here, T cell recognition for antigens formed by genetic recombinations with variable and constant regions. So these are really what they kind of look like. All right. And remember, all right, so these are receptors for the antigen. These don't secrete. Remember, antibodies are secreted by B cells. These are T cell receptors. Remember, I said T cells are hand-to-hand combat. B cells literally pump out antibodies. So they're floating around, they're secreting antibodies. T cells literally, they're like ninjas. They are hand-to-hand combat. Right. So B cell maturation is directed by bone marrow sites that harbor stromal cells. Right. So stromal cells, in case you guys have forgotten, these are cells in your bone marrow. They're like little nursemaids. They just sit there and they give these B cells growth factor and anything they need. Stromal cells actually cause it to develop and mature, which nurture the lymphocyte cells and provide hormonal signals. Right. So if they need to ramp up production, the stromal cells give them hormonal signals, little growth factors, say, listen, guys, I need you to pump up production immediately. 1 million of distinct B cells develop home to specific sites and lymph nodes, spleen and gall. Remember, this is gastro-associated lymphatic tissue. These B cells are just sitting there in your lymph nodes, Peyer's patches, your tonsils. They're just waiting for exposure. So remember, this is a good thing, because if you have, like, I don't know if you guys have your tonsils, or please realize, why would it be important to have B cells sitting in your tonsils, right? Or in your Peyer's patches? So if you swallowed, came across something, these B cells that are in your tonsils they're come across it immediately. They're like little sentinels. They're gonna say, wait a minute, I just swallowed this or just came across this. They send signals right away to dendritic cells. They take them to lymph nodes and go, listen, guys, we just came across X, Y, and Z. We've never seen it before. Do any of you guys recognize this? If you do, make some antibodies. If someone says, oh yeah, I saw that when you were 12, let me get a hold of the plasma cells. They have the recipe. They make a whole bunch of antibodies. Now we have antibody producing factors. Hold on, guys, I got to plug my MacBook in. Hold on one second. You guys are probably saying, thank God, I thought he'd never shut up. Okay. All right. So these come in contact with antigens throughout your life, and they have immunoglobulin as surface receptors for antigens. Remember, if you have these in your stomach, in your Peyer's patches, if you ate something that had a pathogen under bacteria, if you've seen that bacteria before, your Peyer's patches recognize it, and they'll start producing antibodies to it immediately. All right. So I don't know if anyone of you have had food poisoning before. All right. But if you have never been exposed to it before, you can get a serious case of food poisoning. If you've been exposed to it before, hopefully you're going to throw it up. All right. Because, you know, people say, well, I got sick, I threw up. Well, no, actually, you're healthy if you threw up. If you're not healthy and you don't throw up, then you're going to process all that stuff. And go, let's say that you process all that food. If your Peyer's patches pick it up, right, for the first time we make antibodies to it, the second time we can send out antibodies right away. We already have the recipe for it. All right, so T cell maturation is directed by the thymus gland and its hormones. And there's different cases of T cell receptors termed CD, or clusters of differentiation. Now, I mentioned before CD4. Remember, those are your helper T cells. They triage your entire immune system. So they direct your B cells and your T cells. So it directs your B cells to make antibodies and make plasma cells. It directs your CD8 killer T cells to go in and kill any cell that has that specific marker on it. All right, and once again, CD8 cells are hand-to-hand combat. All right, and we also have CD4 CD4 regulator cells. And once again, these CD4 regulator cells, we'll get into that in a little bit. They actually will tell your immune system to calm down. Or remember too, we do want some CD8 cells to attack our own cells if they are virally infected or if they're old and worn out. Right? If they don't want to produce apoptosis, we send a CD8 cell in. We literally go in and kill that cell if it's old, right? Or if it is genetically mutated, the CD8 cell will say, you know what, you don't look quite right. You could be precancerous or cancerous. We're going to go in and kill you. Or I'm going to send my cousin, the natural killer cell, in to take care of business for me. So mature T cells migrate to lymph organs, right? Your lymph nodes, Peyer's patches, or whatever, and they hang out with your B cells. They just kind of play video games until something comes along that they have a lock and key mechanism to, and they wage an immune response to it. Okay, entrance and processing of antigen colonial selection. All right, so antigen, I said before, is a substance that provokes an immune response in specific lymphocytes. And not everyone's going to have a reaction to the same antigen. Some people don't have a reaction to it, and some people, like I said, they may not have a B cell that has a variable region for that antigen. And unfortunately, if that's the case, you're not going to be able to make an antibody to it. You're not going to be able to have an immune response as effectively as someone who can produce an antibody to it. Property of behaving as an antigen is antigenicity. So here's the thing. It has to be foreign. It has to be big enough. It has to be have a molecular weight of over 10,000 molecular weight. It has to have a very specific shape for that antibody. It has to be accessible. Something happened here. And there's things called haptens. So hapten is something very, very tiny, like dust mite and cockroach droppings and cat. It's not really cat hair, it's cat saliva that people are allergic to. It has to actually, that's a hapten. It's so tiny. It has to actually attach to a protein in your body to be big enough to be recognized. It has to have a certain size to be recognized. The body doesn't actually recognize haptens are too tiny. But once you process that cat hair or whatever, your body recognizes it's big enough. All right, characteristics of antigens. Perceived as foreign, not a normal constituent of the body. Foreign cells in large complexes of molecules over 10,000 molecular weight are most antigenic. Right? Antigenic determinant, we have epitopes. Right? So once again, epitope, when I said before in the variable region, epi means on, or small molecular groups that are recognized by lymphocytes. So here's the epitope. And remember I said before that these areas here, they can actually, this Y, you see this little spot here? These can actually pivot, all right? They can pivot and they can change conformation. And once again, you could have this antibody right here could attach to just these. Another one could attach to these. Another one could attach to the red squares. So you literally, on here, you could have 3 different antibodies attaching to this in all different conformations. So this thing would automatically become, theoretically, it would be like 20 times the size it is right now. Your immune system could not possibly ignore this if we had all these antibodies attaching to one of these antibodies attaching to a separate thing here. You'd have 3 different antibodies attaching to every one of these. There's no way your immune system could not see that. So once again, haptens are small foreign molecules that consist only of a determinant group. These are not antigenic unless attached to a larger molecule. All right, so hapten, like rat hair, which is in your peanut butter, by the way, in your hot dogs. Never mind. Carrier groups contribute to the size and complexity and enhance the orientation of the antigen. So once again, cat hair, rat droppings, things like that, they're very, very tiny, but they have to attach to a carrier molecule in your body. Now they become big enough to be recognized. So if you see hapten just realize it's an antigen, but it's too small. They recognize it has to attach to something bigger. So it becomes 10,000 molecular weight in size or bigger. Alloantigens, cell surface markers and molecules that occur in some members of the same species, but not in others. Superantigens, we'll talk about these with some of these bacteria at the end of the semester. These are potent T cell stimulators, provoke an overwhelming response. So what they'll do is these, like staph, some of these other bacteria, they'll start secreting superantigens. It'll actually cause every single T cell in the area to evoke a response. And these T cells will literally, they go absolutely ballistic and they attack everything they come in contact with. They don't pay attention to cell markers. They literally become killing machines. They attack anything around them. An allergen is an antigen that evokes an allergic response. So please realize you could have an antigen, a foreign thing in your body. If your macrophages, monocytes, and your antibodies don't recognize it, it goes through undetected. So an antigen only becomes an allergen if it evokes an allergic response. Autoantigens are molecules of cell tissue for which tolerance is inadequate. All right, this is the problem. All right, and we'll talk in a little bit, and we'll talk about scarlet fever and some of these other organisms with strep, that your body produces antibodies to the strep, which is normal. But the problem is the antibodies in your body think that your endothelium is strep. And if you can imagine what would happen, you have antibodies attaching to your endothelium of your heart valves and the inside of your heart. So imagine what that does. You have an antigen antibody reaction inside of your heart valves. So your immune system says, oh, wait a minute, we have an antigen antibody going on. It's going to send a bunch of macrophages in there to start cleaning it up. It starts destroying your heart valves. Not a good situation to have. Cooperation in immune reactions to antigens. The basis for most immune responses is the encounter between antigens and white blood cells. I just mentioned that before. Lymph nodes and spleen concentrate the antigens and circulate them so they will come in contact with the lymphocytes. Remember I said we want in your reticuloendothelial system, we want pressure to get high enough so that these antigens and these foreign things get into your lymph nodes. We get that pressure up high enough, those lymph valves open up, the fluid goes in. When the pressure equalizes, the valve closes, your muscles can contract and squeezes those lymph nodes like your veins, the one-way valves, to your lymph nodes, right? In there processes those because we said we have a bunch of afferent vessels going in, one efferent going out. So we have a bunch of valves, afferent going in. They start percolating in that lymph node, and we want these lymphocytes to come in contact with them, and we want the B cells to recognize it as foreign, start producing antibodies to it. So antigen presenting and presentation to lymphocytes. I said before, we have antigen presenting cells. Remember, those are your neutrophils. They're not great at it, but they can do it. Major players here are macrophages. Remember, those come from monocytes. Monocytes, when they leave your bloodstream, turn to macrophages. And then in your tissues, we have those dendritic cells. So T cell dependent antigens must be presented by phagocytes, called antigen presenting cells. The phagocytes, remember, these phago means eat, right? Cytes, they go in, they engulf it, they digest it, and they process it on those MHC2 receptors. Remember I said the two MHC2 present these. Antigen presenting cells modify the antigen. Then antigen is moved to the antigen presenting cell surface and bound to the MHC. This should be MHC2 receptor. So they go in, they engulf it, they take the antigen that's specific to that foreign material, that's antigen. They process it, they wrap it up nice and tidy. They present it to the immune system, say, listen, this is what I found. It's foreign. You guys do your thing. Antigen presentation involves a direct cooperation among antigen-presenting cells and a T helper cell. That's your CD4 cell. Interleukin-1 is secreted by the antigen-presenting cell to activate T helper cells. Interleukin-2 is produced by T helper cells to activate B and other T cells. And remember, these interleukin-1 and 2, these also, they tell the T cells to agree it's foreign, and they tell the T cells to reproduce and make memory cells. Interleukin-2 is going to tell the B cells and other T cells it's foreign. I want you to agree it's foreign. B cells, I want you to produce a bunch of B cells, a bunch of plasma cells. I want you to make a bunch of antibodies, and I need memory cells in case we come across it again. And T cells, here's the antigen. I need you to make more T cells. I need you to make memory T cells. And I need you to go out there, and if you see this antigen in any of these cells, I want you to go in hand-to-hand combat. I want you to kill them. Okay, so T helper cell activation, right? So remember, these are foreign microbes. These are foreign. We're going to call these bacteria. Right? The antigen peptides complexes with the MHC2 receptors. Remember, these are transported to the membrane, the surface location of the antigens, and ready presentation of the T helper cell, which is specific for the antigen being presented. So here's your MHC2. Remember, it's engulfing this bacteria. It's literally taking it to the cell surface. It's putting in an MHC2 receptor. Remember, the MHC2 is presenting it, presents the antigen to the T cell receptor. Right? T cell receptor, they're shaking hands here. And I said when that arm comes out and it connects right here, they sign the contract. They say, listen, right, we know it's foreign. Now this T cell here knows that when they come across this antigen on any of these cell markers or anywhere in the area, it's going to go in and destroy that cell. This is very simple. I know it sounds complicated to you guys, but this is a very simplistic. It's a lot more complicated than this, but for this course, it's all we need to know. B cell activation, antibody production. So remember, B cells produce antibodies. So that is going to be in the blood plasma. They produce a chemical. T cells literally are hand-to-hand combat. Once B cells process the antigen, interacts with CD4 cells, and are stimulated by growth and differentiation factors, they enter the cell cycle in preparation for mitosis and clonal expansion. So here's the deal. What they're saying is once the B cell processes the antigen, it knows it's foreign, it talks to the CD4 cell. The CD4 cell sends out that interleukin-1. It tells the B cells in the area, listen, we have a problem. I want you to make more plasma cells because plasma cells are going to make antibodies. And I want you to make memory cells in case this comes up again. We want to be ready with the recipe. We want to be able to make antibody as fast as possible. Divisions give rise to plasma cells that secrete antibodies and memory cells that react to the same antigen later. And it's just showing you here. Here's a B cell. It interacts with the receptor. It sends out that interleukin, the chemical signal. Once again, lock and key. It handshakes, signs the contract. Now it's going to make memory cells for later, and it makes a whole bunch of more plasma cells to pump out these antibodies. Now I think I embedded a video in there. You're going to see once these B cells are activated, once the antigen docks on here and stimulate, this cell will literally pump out like 10,000 antibodies a second. It's amazing. It drops on the cell, it locks in, sends a signal to the inside of the cell like a light switch on and off, and the plasma cell just starts pumping out all these antibodies. They start floating around in your bloodstream. They come across the antigen, the microbe. They attach to it in the blood, and we super tag all these bacteria. Your macrophages come along and engulf it and eat it because it's got this huge marker on it. It's really amazing. for me. You guys probably think it's boring. Anyway, so here's your antibody structure again. All right, this looks very similar to what we saw before. Remember, these are literally, this is the antibody. So we saw before that we saw the heavy chain in the variable region. Those were actually attached to a cell. Right? So I want you to imagine these things are floating around in your bloodstream constantly, floating around in your tissues in your bloodstream. So these are immunoglobulins. You guys have heard of IgA, IgM, IgE, IgG, which mom gives through breast milk and things. So the baby has some immunoglobulins in the first 6 months of life. These, once again, consist of 4 polypeptide chains, 2 of them identical. So these are the same things that are on the cell surface of the B cells, but when the plasma starts just pumping them out, these antibodies literally are floating around in your bloodstream, waiting for antigens. And now, if it's specific for a certain antigen, it'll attach here, right? And now you have a microbe and an antibody antigen antibody complex floating around. The macrophages are seeing this great big things floating around. They go in and engulf it and destroy it. That's how your immune system works. Here's your antigen antibody, right? So if we look, these are really specific lock and key, right? So theoretically, they say that B cells make very specific antibodies to an antigen, right? And it can only be done by antigen-presenting cells. The truth is, it's not in your book, but your B cells, they can actually make they call it like a junk antibody. They can actually make a very genetic antibody on their exposure without being told by a CD4 cell. So it kind of recognizes as foreign. It kind of makes a very generic antibody to it. All right, it's not in this book, but that's what really happens. But for now, just realize. So here's that genetic recombination that your body can go through. It takes a little bit of everything, shuffles it around, recombines it, and then we make antibodies using this genetic code of like 500 different genes. We shuffle it all around and make all kinds of antibodies. We make a bunch of B cells with the hopes that they come across an antigen and make antibodies to it. All right, and here's the antigen antibody reactions I said before. So opsonization, right, is a process of coating microorganisms or other particles with specific antibodies so they are more readily recognized by phagocytes. This is part of the complement system, right? So we go in and we super tag it, right, so the macrophages can't possibly miss it. And neutralization is what we want to do. If we have a virus, right, we want to go in, antibody fills the surface receptors on a virus or active site on a microbial enzyme to prevent it from attaching. So we can go in, we may not be able to kill it, that's T cells do that, but I can go in and I can tag, oh, this is a virus, right? So this is a virus, right there, that's a coronavirus right there, with spikes. If I went in, and this is, the spike is what attaches to the cell, all right, and corona only attaches to pneumocyte type 2. If I go in and I put these antibodies on every single spike, and like I said, I want you to start picturing these like those dryer balls. Next week I'll show you guys one. All right, if I could put an antibody on every single one of those spikes in the virus, when this virus lands on the cell, it can't attach. The lock and key is jammed up. There's something between the lock and key. There's an antibody on it, so it can't attach to the cell. It can't get into the cell. All right. Agglutination. All right, so if I gave you guys my blood, I'm B negative. So there's probably maybe one of you out there who has B negative blood. The rest of you, your body would find my blood foreign, and you'd have antigen antibody complexes floating around. The problem with that is these are great big complexes attaching to the red blood cells, and when these get into your capillary beds, they're going to jam up, all right? So now everything distal of that antigen antibody complex doesn't get any blood supply or oxygen, it'll die. Not good. And if it jams up the glomerulus on your kidney, then you have no filtrate. You'll not make any urine, and your body will increase that blood pressure. It'll actually force through the glomerulus. Now the glomerulus, instead of that filter having little tiny holes, has these you rip great big holes in it. Now all the things in your blood that shouldn't be leaving your blood go into your kidneys, and your nephron has to absorb all that. Causes major kidney damage. So if it doesn't kill you, it'll definitely cause major kidney damage. That's why we cross type blood consistently before we do that. We don't usually make that mistake ever. Complement fixation, activation of the classical complement pathway can result in specific rupturing of cells in some viruses. Not important. Just remember neutralization, opsonization, and you guys know agglutination already. Precipitation, aggregation of particles of antigen. So we know should go in. If we agglutinate it, we make a great big glob of it. Right, we take it out of circulation. Right, and then precipitation. If we have antigen antibody complexes floating around in your blood, what happens is these are so heavy they start settling into your blood, or they can start settling into the joint compounds, and the macrophages come and try to clean it up, and they cause major damage. All right, one of the major causes of rheumatoid arthritis. So functions of the FC fragment. So these are just kind of with the macrophages, neutrophils, eosinophils, mast cells, basophils, and lymphocytes. Right? These are all of your players in your white blood cells for the immune function. Macrophages, remember these go in. These are great big killers. They process things. Neutrophils can process antigen processing cells. Eosinophils, these are going to be elevated if you have parasitic infection or allergic reaction. Mast cells are the cousins of basophils. These are in your tissues, these are in your blood, and your lymphocytes. Remember, your lymphocytes are your T cells and B cells. Certain antibodies have regions on the FC portion for fixing complement. On the FC portion, they actually go on and fit complement, causing major super complexes for the immune system to go in, find it, and wage the attack. All right, so here's your immunoglobulins, right? IgG, IgA, IgM, IgG, IgE. IgE, if it's elevated, that's allergic response. Eosinophils. Right? IgM is huge. Right? This is 5 antibodies all together. This is a great, it has those 5 Ys all together. This thing is major, huge. Right? IgG is produced by plasma cells, the primary response. So this is always the first one on the scene. IgM is second. Right? IgA, now I know I've mentioned this probably the 10th time now. Remember, this is in all of your mucous membranes. Remember I said this 1,000 times? Circulates in your blood. Dimer and mucus and serious secretions. I mean, it's very liquidy, all right? So this is in all of your surface of your body that are on the outside of your body. Right? So you always have IgA in all your mucous membranes, right? IgG is the first immunoglobulin present. This is what mom gives the baby. Right? And IgE, this is another one. This immunoglobulin, if it attaches to an eosinophil or a mast cell, right, it starts pumping out histamine like crazy. All right, so here's the important thing. So everyone here has been vaccinated, I'm assuming, if you're in school, unless you're Amish or you have some kind of religious exception or you don't believe in it, whatever. So what's happening is we have an amnesic response. So your first time you're exposed to something, it takes, like I said, it takes 5 to 12 days to produce antibodies. Here's your secondary response. You've already been exposed to it before. Your plasma cells have made antibodies, or you have memory cells to it. So after the second contact with the same antigen, the immune system produces a major, rapid, stronger response due to your memory cells. That's why you want your CD4 cells to make these antibodies. You want to make memory cells. So now if you get exposed to it again, I said, you already have the recipe. You make these your second response, you make these huge IgM. These are huge. These are 5 antibodies all together. send these out immediately. You send IgG out. These attach to the foreign antigen, and your immune system can't miss it. So this is why we do vaccination. This is why your second exposure to something, you may be exposed to something, you don't even know you're exposed to it because your immune system takes care of it immediately. Right? All right. Unfortunately, flus and colds, they mutate, so it won't be as serious. But if you come across an antigen the second time, you may not even know you have it because your immune system took care of it immediately. I'm going to end here soon because I'm about to lose my voice. All right. So T cells and cell-mediated immunity. All right. Cell-mediated immunity requires the direct involvement of T lymphocytes. So once again, remember, B cells produce plasma. That's in your blood. T cells are hand-to-hand combat. These are cell-mediated. The cells actually attack. Right? I'm going to say that. I've said it about 10 times right tonight. I guarantee you on the exam, 10 of you are going to get it wrong. Remember, B cells are blood, T cells are cell-mediated. Actually, the cells are involved. T cells act directly with antigen and foreign cells when presented and associated with an MHC marker. Remember, I said the T cell, the CD4 cell tells the T cell, here's the antigen. If any cell has an antigen on its cell membrane, if any antigen has an MHC1 or HLA marker on it, I want you to attach to it. It pokes that hole in it, puts that enzyme into the cell, and it kills it, remember, in 20 to 30 minutes. Goes in and literally throws up inside the cell and kills the cell within 20 to 30 minutes. T cell secretes cytokines that act on other cells. Remember, cytokines are cell chemicals. Kine means movement. So these T cells secrete other chemicals. Remember, there's no Wi-Fi, no cell service, so they secrete chemicals. These other cells will pick up the chemical concentration. They realize that it is calling them forward. They follow the concentration gradient towards the infection or the problem. Sensitized T cells proliferate into long-lasting memory cells. So once we activate them, they're long-lasting memory T cells. They last for decades. Right? And like some of these, like rabies and some of these other vaccinations, you have to have it updated every 10 or 20 years because they don't last quite that long, but they're long-lasting. All right, guys, I'm going to end it here. Slide 41. We'll pick this up next week. If you can't make it, I will try to record it again. All right. If you have any questions, just email me. And if you guys don't want to set up an office hour with WebEx, we can do that too. Just let me know. I can go in and put in office hours. We can do a 15, 20-minute slot whenever we can agree on a time. All right. Any questions? Nobody fell asleep. Awesome. All right, guys. Have a good holiday. All right, and I'll talk to you guys later. Bye.
Sounds good. You too. Happy Easter.
Yeah.
See you soon. Lord. Help me, guys. I have no idea what I'm doing here. Ah. Sign out.